module is about the genetic code, and specifically how the genetic code was actually broken by some very classic early molecular biology experiments. First of all, people believed that genes and proteins were collinear, and what that meant was that if you have longer genes that occupied more space along the double strand, the double helix, that they would code for longer transcripts, and then when the transcripts were translated, they would code for longer proteins. So here we see 3 genes of different length along a double helical DNA molecule, transcribed into 3 different length green mRNAs, and then translated into 3 different length polypeptides that have now folded up. So in other words, longer genes encode bigger proteins. So if genes and proteins are indeed collinear, one is free to speculate as follows. Obviously, one base couldn't mean one amino acid, otherwise you would only be able to code for 4 possible amino acids. You'd only get 4 codons. 2 bases, you'll only get 4 squared or 16 possible code words, and therefore you could only code for 16 amino acids. And of course we know that proteins are made up of 20 different amino acids. Imagine that a code word was made of 3 bases, then you could get up to 64 possible amino acid codons. And that was enough to account for the 20 amino acids. That if you had 4 bases per amino acid, that would be 256 different possible codons, and that's way more than enough to get 20 amino acids. And while it wasn't formally excluded, it didn't seem reasonable to go beyond a triplet codon. So now let's talk about how the code was broken. It was Marshall Nirenberg and his colleagues who broke the first few code words by identifying what fraction in an isolate from E. coli cells was actually the messenger RNA fraction. So they took E. coli cells and they burst them open, they lysed them. They got a lysate, and the first thing they did, which was very dramatic at the time, was to add 19 amino acids and a 20th one that was radioactive, along with ATP on the grounds that making polypeptides would probably consume a great deal of chemical energy. And lo and behold, they produced radioactive polypeptides. This was called life in a test tube when it was first performed. It isn't quite that dramatic, but it was the first demonstration of an ability to synthesize proteins, polypeptides, in a test tube. You could then fractionate the E. coli cells into various components, as you know, by cell fractionation. So the first thing with E. coli is to separate at high speed in an ultracentrifuge what was called the microsomal pellet, that is the small bodies of the cell, from whatever was above in the supernatant, a clear cytoplasmic liquid called the cytosol. Then Nirenberg did several interesting things. To the microsomes, he applied an RNA extraction protocol which separated some RNA from the ribosomes, but without disrupting the ribosomes. And then he spun them again in a centrifuge at very high speeds and got another pellet. This pellet turned out to be ribosomes, but without some of the RNA. This RNA from the ribosomes, shown here as fraction 2, was not ribosomal RNA. So the structure of the ribosome was intact, but some kind of RNA had been taken off of it. The supernatant was also extracted, specifically RNA was extracted from the cytosol to get, well, let's call it RNA from cytosol, fraction 3, and then an RNA-free cytosol. So these are the 4 fractions that Nirenberg ended up with. He then did a reconstitution experiment, something we've seen before. He found that if he took the ribosomes and the RNA that had been extracted from ribosomes, added them back together, and threw in 20 amino acids and ATP, the same sort of experiment he did in the first instance, he did not get any polypeptide synthesis. In fact, he tried various combinations, as you see here, and got no polypeptide synthesis until, in fact, he added back all 4 components. At that point, adding ATP as a source of energy, free energy, for this process of protein synthesis, along with the 20 precursor monomer amino acids, he did get polypeptides. He did synthesize polypeptides, radioactive proteins, basically. So it turned out you couldn't leave out any fraction in this experiment. You may suspect, as Nirenberg did, that fraction 2, this RNA that could be extracted from ribosomes but that was not ribosomal RNA, might be messenger RNA. So Nirenberg did a very clever thing. He contracted with a fellow at Harvard to synthesize a monotonous message called poly-U, which is just a string of uridines, uracil plus sugar plus phosphate, UMP, 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 a big string of uridine monophosphates, which is essentially a fake messenger RNA, right? And he added that instead of the RNA that he suspected might be real E. coli message, that's fraction 2. So now he added back ribosomes, fraction 1, RNA from the cytosol, fraction 3, RNA-free cytosol, fraction 4, and poly-U. And one by one, in what amounts to 20 separate experiments, he threw in one of the amino acids and ATP. A polypeptide was indeed synthesized, but only when one amino acid was added, and that was phenylalanine. If the amino acid was phenylalanine, he made polyphenylalanine in this system, from which he concluded that the triplet code word UUU must code for phenylalanine. Nirenberg and his colleagues deciphered several other codons with the other polymononucleotides as well as polydinucleotides and even polytrinucleotides, but that became pretty tedious after a while, and Marshall Nirenberg hit upon a method to decipher all 64 codons in record time. It turned out that if you took cells and you added radioactive amino acids to the cells, and you waited a very short time, and then homogenized the cell, so you end up with an homogenate, and then fractionated the cell into RNA from the cytosol, RNA-free cytosol, and whatever was left of the cell, the rest of the cell, it turned out that the radioactivity, which was radioactive amino acids, was not found where you would have expected it, which is to say they weren't in the RNA-free cytosol. Instead, most of the radioactivity was associated with an RNA fraction from the cytosol, the one we know as transferases. Remember, the RNA from the cytosol was an RNA that Nirenberg had extracted previously from cytosol, so that he had ended up with RNA from cytosol and RNA-free cytosol. Go back a few slides and you'll see that. We know that now to be tRNA. In the old days, by the way, that RNA was referred to as sRNA because it was from the soluble compartment of the cell, the cytoplasm, so it was called sRNA. Shortly after referring to this as sRNA in publications, these RNAs were identified as transferases. So the amino acids that enter a cell rapidly associate with transferases. Before I go and show you how this enabled Nirenberg to break the genetic code in record time, let's go through the aminoacyl-transferase or aminoacyl-tRNA transferase catalytic reaction that associates tRNAs with their amino acids. It happens in 2 steps. The amino acid plus ATP, it's going to provide energy eventually, plus the appropriate aminoacyl-tRNA synthase form an aminoacyl-AMP enzyme complex, liberating a pyrophosphate. So the free energy is actually captured now as this aminoacyl-AMP enzyme complex. Now a tRNA comes in, recognizes this energy-rich AA-AMP enzyme complex, and finishes the reaction by binding the amino acid to itself, that is to produce an aminoacyl-tRNA, liberating the AMP, disconnecting it in other words from the enzyme, and of course regenerating or liberating the initial enzyme itself. So you can sum up those reactions as an amino acid plus a tRNA plus ATP becomes aminoacyl-tRNA plus AMP plus pyrophosphate. Polypeptide synthesis, which we'll be looking at, is one of the most expensive biochemical reactions a cell can do. And one of the first expenditures of free energy is this ATP that has to be consumed to make every amino acid tRNA that's going to be used in translation. Back to Nirenberg. He realized that ribosomes might be induced to associate with synthetic mRNAs as short as 3 bases. In other words, he might be able to show ribosomes associating with literally a codon, a 3-base nucleotide, polynucleotide, a 3-base nucleic acid. And if that were possible, he might be able to show which codon enabled which amino acid tRNA, aminoacyl-tRNA, to attach to a ribosome. So here's the experiment. He found a filter through which tRNAs or aminoacyl-tRNAs would simply pass in solution. Also, individual triplet codons would also pass through this filter. And so he had all 64 triplets synthesized and then isolated an extract of E. coli which contained all the aminoacyl-tRNAs. but he produced all of the triplets and demonstrated that the triplets and the aminoacyl-tRNAs would indeed pass through the filter. He also demonstrated that if he isolated ribosomes, E. coli ribosomes, that they would not pass through the filter, they would be retained in the filter and they would stick on the filter. So the experiment then was to mix UUU and extracted ribosomes with the 20 different aminoacyl-tRNAs. And this could actually be done in a single experiment in a single tube, unlike the original synthetic message experiment. You could mix these all together. Think about why that was possible. Mix them in a tube and then pour that stuff after a moment or two through the filter. The expectation or the hope was, as shown here, that the ribosome would in fact bind to UUU, and if it did, that the phenylalanine-tRNA, shown here, would be induced to bind to the ribosome via the UUU, via its codon. And if it did, then phenylalanine-tRNA bound to the ribosome could not pass through the filter, and the filters could be analyzed to see if there was in fact such a complex. And indeed, that was what was found. So UUU on the ribosome would attract from such a mixture only phenylalanine-tRNA, the remaining codons, were quickly deciphered in the same way. Okay, so that's how the genetic code was ultimately deciphered in short order. And here is the genetic code, all broken for you. So this is one of many ways to represent a table of code words, but let's look at some of the highlights here. First of all, there are 3 code words that did not capture any aminoacyl-tRNAs, and these turn out to be stop codons. These are 3 base codons that would be found near the end of a messenger RNA and would be an indicator for the ribosome to disassociate and fall off because the polypeptide it was making was finished. The stop codons are UAA, UGA, and UAG. I'll show you how you read this particular genetic code dictionary. Watch the order of nucleotides or bases lighting up here in the next click. First there's an A, then there's a U, and then to the right near the top there's a G. That AUG, right? First base is A, second is U, third is G. AUG is the code word for methionine. And as you will learn shortly or perhaps remember, methionine is the first amino acid put into all polypeptides on this planet anyway. So it is the start codon. I think I threw in another one here as an example. UAG codes for tryptophan. So if you needed a little more coaching on how to use this genetic code dictionary, you can look at that. UAG codes for tryptophan. Nothing special about tryptophan, just wanted to show you how you would read the genetic code dictionary for a second amino acid. You can now do that yourself for a few different amino acids. So for example, you can look at cysteine, and you will see that cysteine tRNAs are expected to have an anticodon for cysteine in the so-called anticodon loop. And let's see, the other one down here, the other one is histidine and glycine are shown as examples. You can look at the codon and see that the anticodon is the complement of the codon. So you can do that on your own using this genetic code dictionary or the one in your textbook, which is laid out a little differently, does the same thing. Finally, tRNA. It is the decoding device. It's because at one end it has a site to attach an amino acid, and usually has one at that 3' end. And at the other end of this molecule, when it is folded over, is the anticodon in what's called the anticodon loop. So on the left you see the traditional cloverleaf structure of a tRNA, and on the right you see what emerges after doing X-ray crystallography of tRNAs. It's kind of an upside-down L-shaped molecule. The 3' end is shown here. It doesn't say 3', but because it says amino acid attachment site, that is the 3' end. And again, opposite is the anticodon loop. Take a quick look to the left one more time. You see a number of oddball bases. There's a Greek letter psi, there's a D, there's an M2G, a methylguanosine, there's an I for inosine or inosine. These are unusual bases in the RNA sequence, and these were put there after the molecule was transcribed. We did talk about transcription of different RNAs, including tRNAs. These unusual bases represent post-transcriptional processing, and here you have an example of some quite specific ones. And that brings us to the end of this presentation.